Okay. Well, shall we get started, Susan?
Let's do it.
Okay. So welcome, folks, to the SEFS seminar. I'll just make a couple of opening remarks here. The first thing I want to mention is just some logistics. So you're on a webinar format, so you can see Susan's presentation, but we can't hear you. So if you have questions for Susan, which we encourage, I would ask that you either put those into the chat or put them into the Q&A option, and I'll be monitoring both of those, and then I'll facilitate the asking of questions after Susan's presentation. So that's the logistics. Today we're welcoming Dr. Susan Waters here. I've known Susan for more than a decade, somewhere on the order of a dozen years or something like that. She's the founder and lead ecologist at Quamash Eco Research, a small Western Washington LLC dedicated to ecological research in support of restoration and conservation. Her training is in pollination ecology and plant community ecology with an emphasis on species interactions under climate change. She earned her doctorate here at UW, where she focused on native exotic plant interactions as they were mediated by pollinators and the effects of phenological shifting on those interactions. When she was a grad student, she also co-founded and co-directed the Urban Pollination Project, which was a citizen science initiative here in Seattle that investigated urban land use impacts on bumblebee foraging and urban food production. She now concentrates her research on Pacific Northwest prairie ecosystems, where she works on plant pollinator community responses to restoration and on the conservation biology of rare plant and insect species. So Susan, the floor is yours. I'll keep my video on so that you have an audience, but we'll turn it over to you.
Thank you so much, John, for that kind introduction. Oops. Let's see. Sorry, just one moment here. Having a little moment. Aha, great. There we go. Am I right? Everybody can see?
Yep, you're good.
Excellent. Well, thanks again for having me. It's really fun to come back as a visitor. And thanks particularly to John, who was definitely one of my mentors when I was at UW and who actually introduced me to the ecosystem that I'm going to be talking about today. So thanks, John. I'm going to talk about an ecosystem that many of you may be familiar with, although there's a lot of people in Western Washington who aren't yet. And that's the Willamette Valley, Puget Trough, Georgia Basin prairies. You can see a picture of the beautiful prairies on the screen and a map showing you the historical extent of these prairies from kind of central Western Oregon all the way up through our region and up into the Georgia Basin. And this is where I've concentrated pretty much all of my work ever since I've known John, so for more than a decade. And that's what I'll talk about today. So I'm going to start by moving this little thing out of the way so I can see my slide. I'm going to start by giving you a little bit of an overview of a kind of a conceptual model of how I think about restoration in the system that I work in. And then I'll use that conceptual model as a framework for the rest of the talk. So in the prairies where I work, I think of restoration in these terrestrial ecosystems as kind of an informed, intentional manipulation of more or less three ecological variables: disturbance, propagule pressure, and competition. So we manipulate these things through, things like pulling up invasive species to diminish competition, planting native species to alter propagule pressure. And we do those things, here we go. We do those things with the intention of having an impact, a positive impact on plant diversity and abundance. We're trying to manipulate that plant community. And so I've created a kind of a cartoon plant community here where you see a bunch of blue circles that are exotic plants and green squares and plus signs that are native plants. We carry out these actions in the hopes of transforming that community, transforming the composition of that plant community. And specifically with the hopes of increasing native plant diversity, increasing abundance of rare species, and decreasing the abundance of exotic species, as you see in this picture. So there are fewer blue circles, there are more kinds of green shapes. And when we do this, usually these are our goals. So we're oftentimes the way that goals are expressed in restoration in the system that I work in is in terms of the plant community. Sometimes it's focused on the diversity of the plant community itself, the native diversity. Other times it's focused on native plants as habitat for other organisms. But either way, it's focused on the plant community. Having trouble. Okay, here we go. But when we carry out these restoration actions, one of the things that I'm most interested in my research is looking at both the direct and indirect results of those restoration actions on other organisms. We're rebuilding this plant community at the basis of all of our trophic interactions, right? But everything else is responding to that. And historically, I think that we have, as restoration has developed, we've had this sense that if you build it, they will come, right? We change the plant community and we sort of assume that a lot of other things fall into place. And I'm very interested in that assumption and the subtleties of the outcomes. So the other organisms that I'm most interested in and that I'll talk about today are exemplified in this beautiful painting that a friend sent me recently. This is a bee sleeping in a morning glory. So I'm specifically interested in what happens when we do restoration, both directly and indirectly. What are the impacts on pollinators? And I'll take a moment to just talk about pollination before I go on with the rest of my conceptual model, because I want to explain why I think it's so particularly important to look at the responses of this group of organisms. So pollination is a mutualism, as you all know. You have a plant and an insect. The insect visits the flower. The insect feeds at that flower and hopefully gets nutritional benefits that in turn support fitness, while the plant in turn is also getting a fitness benefit from both receiving conspecific gametes and distributing its own gametes. So if all goes well, it's a mutualism. And because it's a mutualism, it's really highly relevant to consider in terms of restoration. Pollination is really important. The best estimate we have globally comes from Jeff Ollerton and his group in 2011. And that estimate is that roughly 85% of land plants, this enormous proportion of land plants, depend on pollinators to reproduce. And when we go and look at our very own system here in the Northwest and check in like, well, what does that mean for us? We find that it's absolutely true for our system. So you can see all the pictures on the screen come from the prairies that I work in, and the figure in the middle shows you what we find when we compare plants from which pollinators have been excluded so that they can't access the flower, and plants that pollinators do have access to, and we see to what degree the plant can set seed. And what we find is that sure enough, both in our native plants and our exotic plants, we have well over 3/4 of them that are pollinator dependent. And so the upshot of that is that when we look at these landscapes that look beautiful and that we care about, these are landscapes that can't be there unless we have pollinator services. Literally, we just, unless those individual plants are kind of there and they're just gonna, maybe they've been planted and they're gonna last for a while, but then individuals will die. Unless there's reproduction going on, that landscape won't stay. So there's an additional arrow that I'd like to add to this model, and that's the influence of pollinator diversity and abundance back on plant diversity and abundance. And then, as we all know, in ecology, we have emergent properties, right? The whole is more than the sum of its parts. So we have restoration influencing plant diversity, restoration influencing pollinator diversity, And then as we increase the richness of both the plants and the pollinators, we may influence the way they interact with each other, the community structure, who's feeding at who, whose pollen is being transferred by who, and how those interactions are structured. And how those interactions are structured in turn affects function and affects a question that I'm really ultimately interested in, which is how and whether changes in community structure can influence resilience in the future as we experience more environmental change. So this is the overview that kind of covers all the sort of suite of questions that I'm interested in my research. And I'm gonna walk through this model as we talk. I'll start by just giving you a little bit of background on how restoration actually happens in our system. This is not a part I'm doing research on. I'm not researching disturbances, but here, this is just the background for what's being done. So in terms of how we manipulate disturbance and propagule pressure and competition, here's an example of a not very restored prairie. This is Cavness Ranch from, I think, 4 years ago. You can see a person far off in that picture. It's about June. And as you look at that plant community, you can see there's some diversity going on, maybe kind of in the distance, but the majority of what we're looking at here is some non-native grasses and one non-native forb, which is Hypochorus radicata, the kind of dandelion-like plant that you're seeing there. So not very much diversity and very much dominated by these exotic plants. So at a site like that, we might carry out some of the following steps on this slide. Historically, these prairies had a disturbance regime that has been lost, which was a periodic fire. The fire was both lit by accidental lightning strikes and also purposefully lit by Salish peoples. That disturbance was lost when Europeans came to this area. So one of the first things that we do in these sites is reestablish some of that disturbance by carrying out prescribed burns. So we do that, and then the next 4 pictures or so show you native seed, and native seed being put in a seeder and planted to grow seedlings, and then seedlings planted out on the prairie. So we're manipulating propagule pressure by increasing the number of native propagules on the prairie. And then the last picture in the lower right-hand corner shows people spot spraying. They're spraying individual clumps of tall oatgrass, and that, of course, is helping to manipulate competition and competitive outcomes. So we do all that, and we do indeed see an impact on plant diversity and abundance. And I'll show you just in this picture here loosely what we see, which is much greater diversity than I showed you in the previous picture. It's a little unfair because this is also a little bit of a different time of year, but you can obviously see there's a lot of different leaf shapes, there's a lot of flower shapes, there's a lot of flower colors. We definitely see a really strong impact of some of these actions. And if we go ahead and look at the data, we also see that very clearly in the data. So this is data from 3 sites. Bald Hill is in Corvallis, Oregon. Smith Prairie is on Whidbey Island. St. John's Prairie is in Portland, Oregon. These are sites we visited in 2 years, as you can see in the figure. And the y-axis is showing you floral richness. At each site, we were comparing units that were restored and unrestored side by side. And so in every case, everywhere we go, no matter what we look at, there's always greater floral richness in restored prairies. So that's for sure an outcome of some of these actions. And just to be complete about the story, we also see an increased abundance of rare plants. This is golden paintbrush, familiar, beloved plant to many of us. John has worked on it quite a bit. Peter Dunwoody has worked on it quite a lot as well. And so we see higher abundance of rare plants in those prairies. So that's all by way of background. And now I want to talk about some of what we found when we go and actually look at how the pollinators are responding. So first of all, let's look at pollinator diversity and abundance. We definitely see systematically, pretty much everywhere we look, that pollinator diversity is indeed increasing with restoration of floral diversity. On the left hand of the slide is it's a bit of a grab bag of genera. It's a list of both fly and bee genera, and maybe with a few other extra characters thrown in there, just to give you a sense of how many genera we're talking about, not even species, but genera we're talking about when we go and look on the prairie. And then the figure shows you floral richness increasing in response to pollinator richness at restored and unrestored prairies. We see this pattern in the South Sound, we see it in Oregon, we've seen it again and again. So that's really exciting. We're definitely seeing pollinator diversity. Just to give you a sense of what that pollinator diversity looks like, in the lower left-hand corner, you can see a bit of a rogues gallery there. This is just one taxonomic group of bees. This is mason bees. We have a lot of mason bees. The taxonomic keys for mason bees are not, they're incomplete and they're hard to use. So we separate our specimens into morphospecies, and we find a lot of morphospecies, and probably there are more species within some of those groups. So you can see the differences in the faces. They're kind of fun to look at. So pollinator diversity definitely is increasing with restoration of just to take a quick look at who's actually visiting these plants. Again, here are the same 3 sites I mentioned before in 2018 and 2019. And I've just split up the number of flower visitors we observed into very loose guilds or groups of pollinators: bees, beetles, flies, Lepidoptera, wasps, and other, which is a group that's basically ants and true bugs and maybe some things that are not great pollinators. And you can see that most places that we go, the diversity that we're seeing is bee diversity, with fly diversity being a close second. So we think that bee and fly pollination are probably the most important processes in these prairies. And if we zero in on some extremely loose groups of bees, we again see that their abundance is always higher in restored prairies. So I've just divided the bees here into bumblebees, which are native eusocial bees, honeybees, which are non-native eusocial bees, and solitary bees, which is an enormously large grab bag of everything, but at least they share a lifestyle. And you can see again, red is unrestored, red is restored, blue is unrestored, and we see higher abundance of almost everything, almost always, in the restored prairies where the floral resources are, which is not surprising. So we definitely are seeing a positive impact on pollinator diversity and abundance. I want to move for a second to talk back from the plant perspective and zero in on some of our rare plants and what they need or are getting in terms of pollinator services. So here on this slide, I'm going to show you 2 different rare plants. I'll start with the one on the left. This is golden paintbrush again. It's a federally threatened species, possibly to be delisted soon. I'm not up on the latest on that. Castanea levisecta. And the point here is that Castanea levisecta is a perfect example of a rare plant that is super pollinator dependent. In the figure in the lower left, you can see the comparison of the number of seeds golden paintbrush makes when it's open to pollinators versus when it's only able to self-pollinate, and it makes 0. So it's highly dependent. And that's, so again, pollinator visitation has to be an essential piece of making sure populations of this threatened plant can stay established. On the right-hand side of the screen is another plant that I've worked on, actually considerably more, Erigeron decumbens. It's the federally endangered Willamette daisy. And Erigeron decumbens is pollen limited at many of its sites, as exemplified in the figure in the middle there. And what that means is that it is reproducing successfully, but the degree to which it's reproducing would be greater if it were receiving more pollinator services. And in the lower right-hand corner, you can see a scanning electron micrograph of the stigma of a Willamette daisy flower. And there are two smaller pollen grains and then one larger one. The two smaller ones are conspecific pollen grains. In other words, that could fertilize this flower's egg. And the other pollen grain is from a different species. So there's some pollen clogging going on there. So the dynamics of what's happening with pollination are likely quite relevant to Willamette daisy in terms of keeping its populations thriving. Before we return to the model, I want to take a brief little, I don't know, sidetrack off on Willamette daisy. And that's because we have found that not only is Willamette daisy clearly a plant that's going to respond to the pollinator community that comes back during restoration, it is also a plant that itself supports an astonishing diversity of pollinators. I'm going to show you probably half of the pollinators that we have seen on this one plant. Again, this is a federally endangered plant, it's very rare. So here's and I'll also pause and say all of these wonderful photographs and many of the photographs in my talk were taken by the marvelous entomologist and photographer, David Kappert. So many thanks to him. So all on Willamette daisy, here's a series of halictid bees. Here's a bunch of cuckoo bees. We've got some Coleoxys here on the top. Here's a Nomada. Here's a longhorn bee, Melisodes, some leaf cutters, and a carpenter bee. Just a really incredible diversity visiting this flower. And that's before we even start to look at the flies. Here are 3 of the many varieties of syrphid flies that we saw visiting this flower. So it's really supporting a lot of insects. And that was actually kind of an unexpected finding as we started working with this particular plant. So we have pollinator diversity and abundance responding to restoration. We have plant diversity and abundance responding to restoration, including rare plants potentially getting more pollinator services. Then one of the main thrusts of my work is to look at what happens to community structure and function as plants and pollinators interact in response. And so I'm going to move on to that next. I use a tool called network theory to study plant pollinator communities in restoration. And I'll talk a little bit about the reason why I use that tool and then show you what I've done with it. First, I want to just give you the background so you can understand what you're looking at as I talk about these. I'm sure many people have seen them before, but just a brief overview. So as you look at this picture, along the bottom of this figure is a series of black rectangles. Each of those represents a plant that was in flower at the site when we went and collected this data. So those black along the bottom, that's the flowers. Black rectangles along the top represent the pollinators that we saw visiting flowers. And if a line connects the pollinator and the plant, we saw that pollinator visiting that plant. And the thickness of that line is proportional to the number of times we observed that interaction. I want to actually hasten to say, for those of you entomologists who might be zeroing in on the fact that some of these pollinator names look a little lame, this is just a cartoon. We really do identify our pollinators, so don't worry. Okay, so that's how you look at one of these networks. And you can see some things right away looking at this one. For example, the giant black rectangle at the bottom called hyperad, that's a non-native species. And you can see in this picture that we have detected a whole bunch of pollinators only visiting that and not visiting anything else. So that's an interesting theme that I'll come back to later on. I'm going to show you one other way of looking at networks because I'm going to use both ways in my talk. This grid is an alternative way of portraying the same kinds of interactions. In this particular one, the pollinators are across the bottom, the plants are across the left side, and when a square is darkened, that means that plant interacts with that pollinator. The darker the square, the stronger the interaction, or the more frequent the interaction. So I like to use these because they are a community-based approach and they're very visual. You can visualize these patterns of interactions, and it's really helpful both in getting a sense of what's going on at a community level and also in generating hypotheses that directly influence, for example, the rare plants that you're interested in. And lastly, the other thing that I like about using this system is that it allows us to actually analyze some of these networks for metrics that are potentially associated with resilience. And I'll come back to that in a little while. So the kind of data that we collect in order to generate these networks is pretty simple. I'm going to show you the results from a study of 6 sites in South Puget Sound. The sites span 0 years of restoration to roughly 12 years of restoration, depending on how you count. I'll come back to that question as well. And it also includes a reference site that did not lose full, it wasn't grazed. It didn't lose its diversity completely. So we use that as a reference. And the way that we collect the data is we visit each site every 2 to 3 weeks. We assess floral abundance, as you can see in the pictures below. Sometimes that takes a while. And then we perform 30-minute observations of pollinator visits at every single forb species that is in bloom at that time. And that can be a lot of species. We observe 2 patches. We have a minimum patch size to make sure that we're watching enough flowers to be able to attract a bee. And then everything that comes in visits that patch, we collect it. Unless it's a queen bumblebee or a butterfly, and we exclude those for conservation reasons. We try to identify them visually. And then we identify everything, and we're able to feed that into the networks. We've done 4 years of data collection at these sites. Unfortunately, due to some funding issues, we've only just now gotten back results from last year. So today I'm only going to be talking about 2 sites worth of data. So you'll have to stay tuned. But when we collect these data, this is the kind of thing that we see. This is a plant pollinator network from Glacial Heritage Prairie. I've color coded it by the different pollinator characters involved there. And you can see a couple of things right away. You can see that this is a bee-dominated site, although there's plenty of fly action, and that there's not very much Lepidopteran action at all. I'll just pause here and also point out, for those of you who are interested in this kind of thing, we currently have something like 8,000 pollinator specimens from this project, which to the best of our knowledge is by far the largest collection in existence for Cascadia Prairie. So we're hoping that this will give rise to a lot of future work as well. So great, we have these plant pollinator networks. They look pretty, they're complicated, they look kind of interesting, but what do they mean? How can we use information on community structure in restoration and in conservation? Well, I'll tell you a couple of ways. So the first a very applied way. We can ask, for example, are we restoring plants that are supporting diverse pollinators? And the reason why we might want to ask this question is because restoration takes a long time. Restoration in these systems takes more than a decade, and it's iterative. So we're regularly burning, and every time we burn, there's more seeding or more plugging of seedlings. And so we continue to add plants over time. One possibility is that if we identify plants that we know support a wide range of pollinators, it would be useful to know that, because we could make sure to plant those plants early in the restoration process and thereby be supporting pollinator communities that would then be in place when more specialized plants that require specific, very small, narrow groups of pollinators come onto the scene. And we can do this really easily with these networks. I've circled here some plants that you can see that the circles that I've drawn cover plants, again, plants across the bottom, pollinators across the top. The plants that I've circled have a lot of lines coming out of them. They're supporting a lot of pollinators, or they're at least being visited by a lot of pollinators. And so when we do this, we come up with some plants that are really familiar players that this really isn't new news. Camas, Oregon sunshine, and Plectridis congesta. These are all plants that are either we're already planting them everywhere or they're pretty common already. But we also find some less familiar characters showing up, including these two flowers. Potentilla gracilis is on the top there, and Micranthes integrifolia on the bottom. And these particular species turn out to be supporting a range of pollinators that are not as much supported by other species that we're planting. So they're actually shouldering a large portion of the increased pollinator diversity. And so we suggest that that might be important. And they're also, Potentilla in particular is supporting a very, very wide diversity. And so might be a really helpful thing to have early in the restoration process. The other thing that we can do with networks is to identify invasive plants that are supporting a lot of pollinators. And we have some. Lepidium campestre there on the top, Leucanthemum vulgare, and Crepus capillaris, we know are all supporting a fair number of different pollinators. And one question is, how important are these plants to native pollinators? Does it matter? You know, we spend a lot of time, effort, energy, and money to get rid of these plants. So one thing we can do with the networks is we can actually simulate getting rid of those plants and have a decent hypothesis about what might happen to pollinators as a result. And so I'm going to show you a couple of simulations here. So these simulations, as it says on the screen, they use these networks and they remove all of the forbs one at a time and then predict which pollinators are not dead, are not gone out of the system. We're going to just look at a subset of the forbs, so only the invasive ones. So I've organized this so that the first forbs that get extirpated are the ones that are introduced. And I'm going to show you Johnson Prairie. It's a very diverse site. I'm going to extirpate seven non-native forbs at that site. Their codons are shown to you there in the lower corner. And the figure that you're going to see, the x-axis is the proportion of plant species that have been extirpated out of the network, and the y-axis is the proportion of pollinator species left in the network. And we're only going to look at the first 7 dots on the graph, because those are those 7 plant species. And when we do that, what we find is an estimate of about 10% pollinator loss. So 10% is a fairly big number. It's not a huge number, but it's pretty big. Let's compare that with what happens at a site that is pretty depauperate to start with, like a not very restored prairie. It doesn't have as many non-native plants to get rid of, but it also doesn't have as many plants to begin with. And I've done that for Cavness Ranch. Extirpated 4, those are the 4 main non-native forbs at Cavness Ranch. And when we do that, we see an 18% estimated pollinator loss. So this is interesting food for thought, because the conservation implication here is that maybe it's important to provide alternative native flowers before you get rid of all the weeds. So this is a message that I am interested in getting out in general to those who are working on this. It doesn't mean it's not important to continue to work to reduce invasive plants, but we need to pay attention to how we're doing it. And we need to at least contemplate the idea that it could have an impact on pollinators. And that's because pollinators don't see native and exotic, they see food. Now, those of you who have worked with pollinators before, or maybe other insects, are looking at this going, oh my gosh, when is she going to talk about the major caveat? So there's a huge assumption in this simulation. And the assumption is this, that the pollinators don't change their behavior. Well, we actually know that pollinators do change their behavior. They're very flexible. Many of them are super generalist. And in fact, when you take away Hypochorus, many of them are perfectly happy to go feed on something else, to the degree that there is something else there to feed on. So we're very interested in doing some manipulative studies to see what really, how do pollinators really behave when we're getting rid of invasive flowers? This is a pilot study that was done by a student who worked with me, Amber Haley Kinnaird. And she asked, how does pollinator behavior rewire networks when you take away an invasive plant? And so what she did was to go and find some plots that contained Hypochorus, which is the dandelion-like invasive forb that you see there. And she collected network data. You can see an example there. So again, the plants are along the bottom, the insects are along the top. And the biggest black rectangle along the bottom there is Hypochorus. That's the non-native. So she did these observations and she did a before-after study. She collected the network data, and then we went through with clippers and we chopped off every single Hypochorus flower head. You can imagine how long that took, and returned to the plot just 2 hours later after getting rid of the Hypochorus. And this is what the network looked like. So there was some reorganization that happened. We saw connectance of the network, which is a network level metric. We saw that increase. We saw the diversity of pollinators increased for 2 of the native species. That could be good or it could be bad. Could mean those native species are getting more pollen, or maybe they're getting more pollen of various kinds, but definitely it's a change. So this is just a pilot, but it's certainly a really relevant question for what we're doing in restoration. How do the pollinators respond, adapt, react to this kind of thing? So anybody out there at UW who's interested in continuing this work, talk to me. I'd love to get somebody else to continue that pilot. Okay, so talked a little bit about plant diversity, pollinator diversity, and community structure. And I'll go on to talk about resilience to environmental change, and the closing chapter of the talk here. So what I'm going to do before I talk about the network metrics is I'm going to first just show you the 6 sites that I initially talked to you about. These sites are a chrono sequence of restoration. Here is what their networks look like the first year that we started doing the study. So here's a site that has had 0 years of restoration, 2 years, 5 years, 8 years, 11 years, and reference. And this is really cool. This is one of the reasons why I like working with networks. You can just see the complexity. You can see it coming back. You can see things are happening. So that's neat, but just seeing that there are more players interacting with each other doesn't really tell us anything about the actual structure itself. And there are ways, there are a variety of different metrics that can be used to look at that network structure. John, I'm just going to pause here and say, could you remind me of my stop time?
I think if you go anywhere in the next 10 minutes, you're fine.
Okay, great. Well, I'm going to hasten through this a little bit then. So I'm going to just briefly describe 2 network metrics that are in the literature. There's a lot of conversation about what network metrics are relevant and what metrics might be associated with positive conservation outcomes and or resilience. So this is still an area that's in flux, and I'm going to describe 2 of these metrics that we've decided are, that we believe are important. And the first one is called nestedness. I'm going to use the example of this little grid here just to remind you how to look at the grid. A, B, C, D, and E are pollinators, V, W, X, Y, Z are plants. I'm going to show you 2 contrasting networks. This one on the screen right now is a sort of random one. I'm going to contrast that with a nested network. You see that on the right. In a nested network, almost all the players have sets of partners which are subsets of the most partnered players. So in other words, Z, Y, X, and W plants all have sets of partners that are subsets of V. Okay, that's a nested network. Nestedness can be thought of as being positive for stability because you can lose a lot of species, those outer edge species, without compromising the core of interactions. However, nestedness is also associated with supergeneralists. And so as we look at deeply invaded sites, we often see increasing nestedness. And that greater sharing of partners also generates more competition, which renders parts of that network unstable. So for nestedness, I'm going to make the claim that what we're looking for in conservation is actually less nested networks. The other metric I'll talk to you about briefly is called modularity. Again, we're comparing two networks here. The left one is random, the right one is modular, and all that means is that there are small subsets of plants and pollinators that are interacting a lot with each other, and they're only loosely connected via one or two players to other modules. Modularity is considered positive for resilience because modules can be lost. You can actually lose whole groups of plants and pollinators and still have a functioning entity in the form of the other modules. And modularity also reflects a fair amount of specialization and diminished competition. So we're aiming for to look and see whether our restoration actions are reducing nestedness and increasing modularity. So just to return to the overarching question, can restoration increase plant pollinator community resilience? Our null hypothesis, well, we know restoration is changing these networks, but maybe it isn't increasing these particular metrics that we care about. The alternative is that maybe restoration really is altering network metrics that are associated with resilience. Here are those same hypotheses put into visual form. On the x-axis is sort of an imagined progression of restoration from unrestored to late stage restoration. On the y-axis, you can see modularity increasing with restoration, and you can see nestedness decreasing with restoration. These are the hypotheses. Now, the x-axis here is looking pretty loose, and I'm gonna punt on that for a little while. I am gonna show you some results. On the x-axis of the results I'm gonna show you is floral diversity, which is strongly correlated with how much restoration has happened. We're still working on developing a restoration index based on actual burning and seeding that we will eventually have on the x-axis. But for now, the figures I'm going to show you use floral diversity as a proxy for restoration. And here are our results. So on the left-hand side here is modularity in response to floral richness. Red is 2017's response, blue is 2018's response. And so we see potentially, we see a relationship in 2017, but not 2018. So you have to stay tuned. With nestedness, we do see it going down in both years, and those slopes are both significant. So this is exciting. We have 2 more years of data to come at you soon. So stay tuned. But the bottom line is that the message is looking pretty hopeful. It's looking like the restoration actions that we're carrying out here are really altering the structures of these networks in ways that are potentially very positive. So again, stay tuned. So that's the last bit of data I'll talk about. I just wanted to put this image back up one more time to kind of return to that overarching view. I think it's really important for those of us working in research and restoration to return again and again to these conceptual models of what's happening and you know, influences and mutual influences on the organisms that we care about. Hopefully we'll get to better and better approximations of answers to questions about resilience. And just to close out, I'll just briefly touch on our future directions. So we have plant pollinator network data now stretching kind of all up and down the latitudinal range of these prairies. So we're interested in eventually, hopefully being able to pull out some of the latitudinal trends from that, or possible climate related trends. We're also starting to look at floral traits and insect traits in our network and how those traits influence the organization of the network, both in restored and unrestored sites. We also have many of you may have heard, we have an invasive bumblebee that's arrived in the northwest corner of Washington and is expanding quickly. And we're very interested to see the outcome when that invasive bumblebee reaches our sites, how that will influence the structure of these plant pollinator networks. And of course, we're also always going to be interested in continuing to use networks in whatever ways we can in relationship to conservation of endangered plants and insects, that should say. So always interested in talking to other people who are interested in these same ideas. So let me know if any of these projects is interesting to you and related to what you do. So with that, I'll just give thanks to the many, many people and multiple funders who have contributed to this project. I've highlighted a couple of people's names because they've worked with the project for multiple years, and the lower left-hand corner shows people who've done all the ID of our specimens. So many thanks to all of these people, and I'll happily take your questions. Thanks so much for your attention. It's hard to pay attention on Zoom, so thanks a million for hanging in there.
Thank you, Susan. And so you've got your contact information there. If folks want to reach out to you, they should do that by email would be the best way.
Yep, email would be best.
Okay. So folks, if you've got questions, feel free to add them to the chat or to the Q&A. And we have one from Peter here. Have you looked at the pollinators in terms of their origin, native or exotic?
Great question. That's a really great question. No, but it's a complicated thing to do. First of all, because the taxonomy is so, we spend incredible amounts of time, even with real taxonomic experts, trying to figure out how many species we have. There are lots of these taxonomic groups where there aren't even good keys. This is particularly true of bee genera. There are actually quite a few bee genera where there aren't good keys. So if you don't even know what species you have, sometimes it's hard to even know, like, whether something is native or exotic. The best of my understanding is that the majority of what we have is native. There are a few exceptions to that. We have some drone flies, which are definitely cosmopolitan and probably exotic. We have no non-native bumblebees yet. We have very few honeybees, actually. We do find them in Oregon, but it's surprising how little presence non-native honeybees have at our sites. In terms of solitary bees, I'm just not sure. I'm not sure, but my strong sense is that most of them are native.
So if you took out the ones that you know are exotic, like honeybees or some of these drone flies, would that change your connectivity or anything like that, do you think?
No. Well, that's actually good. That's a good question. Very unlikely, except maybe with drone flies. Drone flies might be an exception. So it's interesting. Different insect behaviors contribute very differently to the structures of these networks. So for example, drone flies, we have Aristolis tenax, we have Aristolis arbostorum. They're probably terrible pollinators, but they're everywhere. And they land on a flower for less than a second oftentimes, and then they zip away, and they come back and they zip away. And so we record all contacts between an insect and a floral reproductive organ. So we count those as visits. But that is definitely an example of an animal that is connected to almost everything. So it could actually influence our outcomes. But I would also say that we see more of them in the less restored sites. So there can be an interaction going on that way too. One of the sites that is the least restored has a high number of flies, like lots more flies than the other sites. And those flies are typically behaving in the way that I just described. So that may be a mechanism that's actually influencing the patterns that we see.
Sure, yeah. How does landscape context relate to pollinator diversity?
Oh, that's a great question. Oh, it's a really good question. So we're of course not able to control much about landscape context. And our sites definitely differ in landscape context, and that may affect our results. Most of our sites have at least some forested area along their margins. And, but it's variable. Some of them also have, well, one still has a working Christmas tree farm along one margin, for example. So there's a range of things. And I would say that that's a question we have not explored really as much as we'd like to. We've done sort of simple things in a non-experimental way, like, go walk along the margins of the woods and see who's visiting the same flowers there near the margins as the center of the property. And we do see some changes, some differences. We do see that even solitary trees in the middle of the property that are shading flowers often cause those shaded flowers to last longer. Their phenology is different. They grow larger, they probably have more water, and they get foraged on in a different way by the bees that we have, particularly bees. And we actually exclude them. We don't watch things that are shaded for that reason, because we think that that's actually probably a salient variable. So that's, to get back to your actual question, I think it's very likely that the context matters, but we have not pushed in that direction.
Okay. We don't have any other questions coming in here yet. Oh, here's one about butterflies. Butterflies don't show up much on your figures. What use are they? Or are they mostly just nice to look at? And if the latter, are they deserving of all the money and attention they get as a focus of conservation?
Oh, come on now. You're asking me to take a philosophical stand here. I've actually been amazed at how few butterflies show up in our data. It's amazing how few butterflies show up in our data. We see them going by, but it's incredibly rare for them to land and actually be nectaring in a patch of flowers that we're watching. I'm not really a butterfly biologist. I've done some work on Taylor's checker spot, but I suspect that I'm reaching a little bit here, but what my instinct is that part of the reason that we don't see them very much in our data is really for two reasons. One, their abundance is low, and two, they're really spending their time looking for host plants. They're looking for places to oviposit. They're not spending a ton of time nectaring. We just don't see that with any of the butterfly species. So as far as what good they are, geez Louise. I mean, they're consumers, they're specialists, they're specializing on interesting host plants. Yeah, they've got awesome meta community dynamics. I'll leave it at that.
All right, so you do see a role for them.
Oh, definitely, yeah.
All right, here's another question. Have you seen a lag between increasing floral diversity and pollinator diversity?
Great question.
And then along with that, do you see pollen limitation in any of the forbs in years soon after restoration, other than the rare species that you mentioned?
Oh, great. These are really good questions.
Two aspects there.
Okay, so the first question is there a lag time? We actually don't detect that lag. I thought there would be. In fact, my original hypothesis was that we would see a sort of U shape where the least restored sites would have such and such pollinator diversity. There's obviously a starting pollinator community that's become accustomed to that locale, right? So you'd have a starting pollinator diversity, and then you go through this burning and spraying and all this stuff that really bottoms out floral diversity, you would think. We actually we haven't detected a U shape in the pollinator response. It just seems to go up. So that's interesting. And could you repeat the second part of that question?
Do you see pollen limitation in any of the forbs in years soon after restoration, other than the rare species that you mentioned?
We do, yeah. Pollen limitation is really common, and it's hard to disentangle. It's hard to disentangle multiple reasons for it. So one thing that happens is that our pollinator populations are booming and busting and changing all over the place independently of the actual the restoration actions and the floral diversity. They're responding to other things besides those things as well. And so that has made it hard to really, yeah, we don't have good data that can say, yes, there's pollen limitation because of X restoration action. But one of the important things that has come out of this is actually realizing that we need to take into account these massive changes in population booms and busts. So the first year that we did this, we thought that we had a system where flies were just as important as bees, because we had as much or more fly diversity and abundance as bee diversity and abundance. The following year, almost no flies at every site. So these huge changes are interesting to track too, again, because flies and bees have different ways of interacting that in turn will influence those networks.
That should also then have implications for your expectations for connectivity or for the network theory and the hypotheses you generate in terms of things can't be too specialized if the specialists are going to fluctuate hugely from year to year.
Right, right. And so I'm super eager for the next 2 years of data for that reason. But I think that what we're going to see, well, maybe I shouldn't speculate. I'll wait.
Okay, leave us hanging. So another question, have you considered different climate change scenarios and how that affects this work and future conservation planning?
Yeah, also a great question. Yes, I have. And so have others. One of the mechanisms that I'm most interested in, but haven't been able to test yet, is relationships between soil moisture, nectar volume, and pollinator foraging. So plants, when they are drought stressed, stop provisioning their flowers with nectar because it's just a waste of water. And on hot days and dry days, we also see foraging goes down. We have an envelope of temperature, humidity, and wind in which we carry out our observations, because when the humidity goes too low, the foraging stops. So knowing that these prairies are likely to get drier and drier summers, that suggests that we're going to have more and more situations where plants are offering less and less nectar. That's the mechanism that I am currently most interested in as far as the direct impact of climate change on the system. There's also, of course, there's things like phenological impacts, but phenological impacts at the community level are really hard. It's really hard to detect a signal, right? One thing is blooming earlier, one thing's blooming at the same time, one thing. So when you have many, many plants and many, many generalist pollinators, it's hard to know what's going to come of that. So at the moment, I'm most interested in the soil moisture nectar relationship there.
At the beginning, when we were chatting, you mentioned that you saw a bumblebee the other day. What about that kind of flip side of how these patterns might change in the winter, early spring, late fall, in that season?
I'm just speculating now. I mean, I guess the best I can do to speculate about that is to say, I suspect that we sometimes have in mild winters had situations where bees basically come out early and can't really find much to eat and don't make it. That is purely speculative, but that is what a bunch of us sort of independently arrived at as an explanation for what happened in 2017 when we saw so few bees. Because we had one of these springs where it was really cold and then it got really warm, things came out, and then it got really cold again. Everything disappeared. And then there was a very long lag time before later in the summer we saw a bunch of other bees show up, right? So that's not data, that's pure speculation. But that kind of weather pattern, you could imagine having an impact like that.
For sure. Yep. Cool. All right. Well, thank you very much again, Susan, for a very interesting talk, and you'll keep us hanging for looking for the additional data when it comes through. But thanks again for joining us over Zoom and for sharing about this really interesting research with us.
Well, thanks to everybody. Really appreciate your time and attention.
All right. That's all for now, folks.